Assalamualaikum. Today we are going to start another lecture that is on the mutation. Mutation is the change or alteration of the in the genetic material that is called mutation. Mutation is the alteration or the change of the nucleotides sequence of the genome of an organism in a virus or extracellular chromosomal DNA. Change in the genome or the genetic material is known as mutation. Viral genomes contain either DNA or RNA, as they may contain RNA or DNA, but eukaryotic organisms may contain DNA only. So, the mutation in the viral genome, either which is RNA or DNA, is the genetic material in that virus case. Mutation results from the error during DNA or viral replication, mitosis, meiosis, or other types of damage to DNA, such as pyrimidine dimers, caused by the exposure to the ultraviolet radiation, which then may undergo error-prone repair, cause or error during other forms of repair, or cause an error during replication, translation, synthesis. During the synthesis of the DNA to RNA, DNA to protein, that is translation, any error from these like pyrimidine in the change in the another form, there is any mutation or change leads to the mutation. So ultraviolet radiation here, exposure to ultraviolet radiation is the main cause of the change. So the UV rays are act as a mutagens or the cause of mutations. Mutations may also result from the insertion or deletion of the segments of the DNA due to the mobile genetic elements. So mutation may or may not produce discernible change in the observable characteristics like phenotypes of the organism. So mutation may lead to the change in the phenotype. Phenotype is the physical appearance. The change in the physical appearance is called mutation. A mutation play an important role in both normal and abnormal biological processes, including evolution, cancer, and development of immune system, including junctional diversity. So mutation is the ultimate source of all the genetic variations, providing the raw material on which evolutionary forces such as natural selection can act. So natural selection may also affect by the mutation, and that is evolutionary point of view may lead to the change in the next generation. Mutation can result in many different types of change in sequences. Mutation in genes can have no effect, alter the product of gene, or prevent the gene from functioning properly or completely. It may change. So mutation may leads to the change in proper way or in a complete change, or maybe in a single trait may change. As a phenotypically we observe like change in the color of the any organism, change in the shape of any organism, length or width of any organism change, that is the phenotypic mutation type. Okay, mutation can also occur in non-genic origins due to the damaging effects that mutations can have on genes. Organisms have mechanisms such as DNA repair to prevent or correct mutations by reverting the mutated sequence back to its original state. So mutation on overview. If we take the overview, what is the mutation? Mutation can involve the duplication of the large section of DNA, usually through genetic recombination. If there is genetic recombination occurs, the change leads to the mutation. These duplications are a major source of raw material for evolving new genes, with tens to hundreds of genes duplicated in the animal genomes every million years. Most genes belong to larger gene families of shared ancestry detectable by their sequence homology. Novel genes are produced by several methods, commonly through the duplication and mutation of an ancestral gene by recombining parts of the genes to form new combination with the new functions. So many years after a long time, there is mutation or a change in the gene occur leads to the mutation that may occur several years after, and function may also affected by the of the organism may affected by the mutation. Here protein domains as such act as modules, each with a particular and independent function that can be mixed together to produce gene encoding. New proteins with novel properties are formed. So with the mutation, the novel properties with the unique properties, new genes are formed. Like for example, the human eye uses 4 genes to make the structures that sense light, 3 for cone cells or cone color vision, and 1 for the rod cells or night vision. So here we also know about this. From these 3 for the cone cells and 1 color for the rod cell for the vision of the eye in the night. All the 4 arises from the single ancestral gene, but if the change may lead to the mutation, and the variation may occur from the one generation to the next generation or the in organism. Another advantage of the duplicating a gene or even an entire genome is that this increases engineering redundancy. This allows one gene in the pair to acquire a new function while the other copy performs the original function. Other types of mutation occasionally create new genes from previous non-coding DNA. So entire gene may also change, may also lead to a change in the phenotypical way, but in the same way may lead to the change in the somehow trait in the living organism or in a living organism, especially in the DNA. Changes in the chromosome number may involve even larger mutations, where segments of the DNA with the chromosomes break and then rearrange. For example, in a human eye, 2 chromosomes fused to produce human chromosome 2. In a human eye, there is a fusion of the 2 chromosomes leads to the human chromosome number 2. That is also the example of the mutation. And the number of large mutation occur here, and the number of segments may change, and the mutation may occur. This fusion did not occur in the lineage of the other apes, and they retain these separate chromosomes. In evolution, the most important role of such chromosome arrangements may be accelerated to the divergence of the population into the new species by making population less likely to interbreed, thereby preserving genetic differences between these populations. So the genetic makeup remain same in the previous, but with the change leads to the into the next one. Sequence of the DNA that can move about the genome, such as transposome, make a major function of the genetic material of plants and animals, and may have been important in the evolution of genomes. For example, more than 1 million copies of allogene sequence are present in the human genome, and these sequences have now been recruited to perform functions such as regulating gene expression. Another effect of these mobile DNA sequences is that when they move within a genome, they can mutate or delete existing genes and therefore produce diversity. These are the examples. This is the example of the allele sequence. The change may lead to the next one and produce a genetic diversity, and with a change in the DNA sequence may leads to the mutation. And for example, we take non-lethal gene mutations. What is meant by the non-lethal? Accumulate within the gene pool and increase the amount of genetic variation. The abundance of some genetic changes within the gene pool can be reduced by the natural selection, while other more favorable mutation may accumulate and result in adaptive changes. Adaptive changes may occur, but the non-lethal change leads to the large amount of the genetic variation in the living beings, and change in may lead to the mutation, cause of mutation. For example, in a butterfly, we could take example of the non-lethal mutation in the butterfly may produce offspring with a new mutation. So majority of these mutations will have no effect, but one might change the color of the butterfly's offspring, making it easier or harder for predators to see. If this color change is advantageous, the chances of the butterfly surviving and producing its own offspring are little. So it may be a favorable, non-lethal and favorable mutations. Example is the butterfly's wings color may change. Okay, now and better with over the time, number of butterflies with the mutation may form a larger percentage for the population. So neutral mutation is another example. For example, we take neutral mutations are defined as whose effects do not influence the fitness of an individual. These can increase in frequency over time due to the genetic drift or a genetic change. It is believed that the overwhelming majority of mutations have no significant effect on the organism's fitness. Also, DNA repair mechanisms are able to mend most changes they become permanent mutations, and many organisms have mechanism of eliminating otherwise permanently mutated somatic cells. This is a neural mutation, and time to time change and permanent mutation type is, and that is not affect the fitness of an organism, so it's may somehow beneficial in a sense. Beneficial mutations, there are some mutations like mutations in the causes. What are the causes? Mutagenesis is the process by which the genetic information of an organism is changed, resulting in the mutation. It may occur spontaneously in nature as a result of the exposure to the mutagens. Mutagens are the substances that cause the mutagenesis or mutation. It can also be achieved experimentally using laboratory procedures. In nature, mutagenesis can lead to the cancer and various heritable diseases, but it is also a driving force for evolution. So what is the main cause? There is a mutagens that act on the organism and organism's genes and may lead to the change. That is may also cause of evolution in the nature and spontaneously may occur. For example, one 2017 study claimed that the 66% of the cancer causing mutations are random, 29% are due to the environment. The studied population spanned 69 countries and 12% are inherited. Only 5% are inherited, 66 are random in the cancer case, or in 29% from the environment, and 5% only are from the heritage. Humans on average pass 16 new mutations to their children, but father pass more mutation depending upon their children with every year, adding 2 new mutations to the child, 2 new changes in the child. So father is more change causer, cause the change in the mutation to the next generation. 4 classes of mutations here are spontaneous mutation, error for prone replication, bypass errors introduced during DNA repair, induced mutations. So what is the spontaneous mutation occur with the non-zero probability. even given a healthy uncontaminated cell. Naturally occurring oxidative DNA damage is estimated to over 10,000 times per cell per day in humans and 1 lakh times per cell per day in rats. Spontaneous mutation can be characterized by the specific change. Naturally occurring DNA oxidative reaction, that oxidative change, that is DNA damage 10,000 times in humans and in 1 lakh times in a rat per day may occur. That is spontaneous mutation. That an example of a spontaneous generation is tautomerism. In a tautomerism, keto form depyrination occur, deamination occur, methyl cytosine in a methyl cytosine mutation may occur, slipped strand pairing occur, replication slippage may occur. So in a keto form, in a tautomerism is also, we have already studied about tautomerism is the in the keto or keto group there is a change in the in the tautomerism a base is changed by the repositioning of the hydrogen atom, altering the hydrogen bonding pattern of that base, resulting in incorrect base pairing during the replication. Tautomerization of the bases, whereby bases spontaneously rearrange themselves to alternative form can occur. So tautomerism is the alternative form formation is called tautomerization. Tautomerization, in for example, in we take in the molecular level, keto forms COO, C double bond O forms a guanine and thymine can tautomerize into ethanol. For example, ethanol or enol OH form. So amino form NH2 forms of adenine and cytosine can result in the rarer amino forms. Double bond NH is formed. That is also an example of the tautomerization. And depyrinization, losses of the purine or G to form the AP site. Deamination may leads to the hydrolysis changes a base to the atypical base containing a keto group into the original amine group. Now CU and AHX hypoxanthine, which can be corrected by the DNA repair. So 5-methyl cytosine T, which is the less likely to be detected as a mutation because thymine is a normal DNA base. Slipped strand mispairing, denaturation of the new strand form of the template during replication followed by the renaturation. This can lead to the insertion and deletion. So slipped strand mispairing leads to the slipping or deletion or insertion in the gene pair or genome. So replication or slipping, in this slipped strand mispairing occurs when error prone. There is increased evidence translation synthesis may occur, template may change, error induced during repair there may error occur, that is double stranded break and then lead to the often a mutation and non homologous joining may occur. So breakage may repair induced mutation can be occur by the environmental causes or mutations induced occur by the chemicals hydroxylane hydroxylamine base analogs, alkylating agents that form DNA ducts or DNA intricating or crosslinks. Oxidative damage may occur. Radiation, how radiation may lead to the change by the for example by cytosine thymine are vulnerable to radiation and they may get changed by and their pairing may get changed, and ultraviolet oxidative damage to the DNA occur leads to the Radiation mutation and ionizing mutation may by the gamma radiation may cause cancer or death. Classification of types of mutations by the effect of structure. By the large scale mutation, small scale mutation occur by impact of the protein sequence, how the by the effect of function, by the effect of fitness may occur. Also mutation occur by the effect of structure. By the effect of structure may if there may be an structure may change leads to the mutation. Large scale mutation leads to the amplification in the non-homologous or homologous, and multiple copies may change or increase in the size and length of the genes may cause amplification. Deletion, chromosomal rearrangement may occur. There is a translocation, change in the homologous chromosome or interchange occurs. Inversions may leads to the also large scale mutation. Loss of heterozygity may occur in a single chromosome. So deletion, duplication or inversion takes place. Then insertion is another phenomena, and translocation may also we can see in the diagram. These the 5 types of mutations we can see here. The for small scale mutation include in the change in the nucleotide. There may be insertion takes place in the nucleotide splice or splice site mutation or frame shift mutation occur, and excision of the transposable element may lead to the insertion or small scale mutation. Deletions occur maybe nucleotide deleted from the any gene site, or substitution mutation may leads to the transversion. And here these are the examples. By impact of the protein sequence, there may also change it takes place. These are here we can see protein coding gene may occur. Transcription occurs to the lead to the lead to the mutation by the pseudogenes or retrotransposons. Here transcription, translation, and we can see here modify post transcription modification. Simply we can say central dogma. Frame shift mutation is also another type of mutation. In here point is substitution mutation occur, or synonym mutation occur. Non-synonym or missense mutation may also occur. Nonsense mutation by the effect of function may also inactivating the mutation. Loss of function mutation, loss of null allele may occur and cause this type of mutation, and that is called haplosufficiency in the normal gene. And if the loss of function may occur or by the loss of function occur haplosufficiency. Gain of function may activate the activation site, so leads to the hypermorph increased gene expression. Dominant negative mutation may lead semi-dominant phenotype are formed. Hypomorphs are also formed. Mullerian classification. Pneumomorph lethal mutation may occur by effect of fitness. We can see deleterious mutation, beneficial mutation of the neural mutation occur here, and nearly neural mutation. But if the see by the inheritance in the reproductive cells, their germ lines may affect, and acquired mutation may occur, so leads to the somatic and somatic mutation, and transmitted to the descendants. Diploid in the diploid genomes of wild type leads to the wild type or homologous non-mutated organisms are formed by them is one of the in which allele mutation occurs. If we see in this heterozygous or homozygous also occur, or compound heterozygous may also occur. Germline mutation may lead in the reproductive cells of that individual, may constitute in the to the offsprings, and from the either parent if occur by called de novo mutation and somatic mutation may passes to the in the somatic cells and transfer to the next progeny by the help of mutation, and that is in UV rays causes cancer, and special causes. Conditional mutation में कर permissive and non-permissive mutation rate is the very rate or time may change with the investigation 50 to 90, and mutationism is the several alternative evolution by the natural selection may occur, and that is led to the macro mutation by the scientist, and proposed his theory, and working after the Thomas Hunt Morgan and other work on that in 1918 may lead to the Darwinism theory. Mutation may natural selection also occurs.